Dear students, welcome to Tungal online classes. So today we'll continue with level 2 questions, right? So level 2 questions are nothing but NEET level questions will be there. So that's why, so let us start with the first question now. So what is the first question? The term cytology was coined by Robert Hooke, Hardwick, Robert Brown, Parkinson. These are different scientist names are there. Among these one, so Hardwick is the name of the scientist, the one who coined the term that is cytology. Cytology is a branch of biology which deals with the study of cell structure and its functions, right? So next question is, who proposed the term cellulae? So it is the first scientist, the one who found the honeycomb-like structures, that is hexagonal structures in the slices of the cork itself. He is none. So who is that? So he is Robert Hooke is the name of the scientist, the one who found the cells for the first time, and he called them as cellulae. Cellulae later become, so what? Cell itself. Cellulae means compartment. So next one is, metabolically active cells have, what kind of structure do they have? So how they are? So is the question arises here. See here, metabolically active cells have similar in size, smaller in size, actually it is smaller in size, elongated form, high surface volume ratio, and all the above. So these are the options here. So usually they are very small in size, and these cells have highest surface area. So as you know, small intestine is there, inside which the surface area is large, but it is made very short due to the presence of many finger like projections called as villi. So they increase the surface area of the intestine. In the same way, so highest surface area volume they should have. Right, all these characteristic features are the characters of metabolically active cells. That's why answer is all the above is the answer for this question. And the next question, bacterial inclusions are generally covered by, see here, bacterial inclusions are nothing but they are the cell organelles, means inside which there are some structures are there. So whether they are covered with the membrane or not is the question. So if it is covered with membrane, then what kind of membrane it is, whether it is unit membrane or non-unit membrane. So the question arises. See the options, let us see here. So non-unit membrane, unit membrane, lipid membrane, no membrane. Of course, some of the organelles do not have any membrane, that is ribosomes are there, of course. But here the question is regarding the inclusions, those which are uncovered with the membrane. So what is that membrane? It is non-unit membrane. It is the non-unit membrane that uncovers the bacterial inclusions. Right? And the next question. See here, prochromosome. What is this prochromosome? The question arises. Prochromosome is nothing but it is another name for nucleoid. Right? So prochromosome is nothing but it is another name. Its another name itself is nucleoid. That's why. So the answer is nucleoid or naked DNA. It is also called as prochromosome. It is also called as. Next one. The basal body of gram-negative bacteria has rings. Already the basal body of the gram-negative bacteria I have showed you there yourself. So you can see, so there one rod-like structure will be there. Cylindrical rod will be there at the center, and that is uncovered with some of the rings. They are 4 in number. So they are arranged in such a way that, so in what way they are arranged and what are the names of each ring. So that is the thing what we are considering over here. So that is MNOP, it is not. EFGH, no. SMLP is the correct answer for this question. So these are the rings. These are the rings that are present in the basal body of the bacteria, that is gram-negative bacteria itself, right? So and the next question is, what is the next question? Pili occurs in. So most of the cases pili are occurring in. So what kind of bacteria they are usually occurring in? So gram-negative bacteria, gram-positive bacteria, all eubacteria, archibacteria. All eubacteria means all true bacteria they are present in, but it is not possible. Archibacteria, they are primitive bacterial cells, that's why there is also no possibility. So here gram-negative bacteria have, right, pili. Pili are usually present in gram-negative bacteria. And the next one, so tubule of endoplasmic reticulum present inside a plasmodesmata is. See here, there is a tubule. There is a tubule present in the endoplasmic reticulum. Endoplasmic reticulum has a tubule, right? So already one thing I have cleared in a theory that this endoplasmic reticulum that mingles with the plasma membrane, which uncovers the entire cell in case of animals. So the interconnection between two cells. So as you have seen, so during the interconnection, the plasmodesmatas are usually quite naturally present inside. So what? They are present inside the plant cells. Through those one, there is a tubule that passes, that continues the endoplasmic reticulum of one cell to the endoplasmic reticulum of the next cell, and that tubule itself is called as desmotubule. Clearly I have said, right? So what is that? Desmotubule is the correct answer for this question. It is not microtubule. It is not cisterna. Cisterna itself is cisternae are there, tubules are there, as well as vesicles all together form endoplasmic reticulum, as well as Golgi complex. It is quite common for both of themselves. Cristae are nothing but they are the finger-like projections, right? So they are the finger-like projections that are made by the inner membrane of the mitochondria. That is why cristae is also not the correct answer. So here desmotubules itself is the correct answer for this question. Next one. Cyclosis or cytoplasmic streaming was discovered by, right? Cytoplasmic streaming in the sense that, so the cytoplasm that moves across the cell itself. So the movement of the cytoplasmic watery substance, that is cytoplasm, that take place across the cell is called as cytoplasmic streaming. It is called as. It was first discovered by one of the scientists, that is Amici. Amici is the correct answer for this question. And other scientists, they have discovered different things. Okay, organoids are. What these organoids are? Already in one of the questions I have solved this one. Organoids are nothing but cell organelles. They are nothing but they are the cell organelles. That is why, so the answer for this question is option C, that is cell organelles is the correct answer for this one. And next one, GERL is present part of endomembrane system, which does not include. See here, already I have told you endomembrane system what it includes. It includes four structures. One is called as endoplasmic reticulum, second one is called as Golgi complex, third one is called as vacuoles, right? So Golgi complex, endoplasmic reticulum, as well as vacuoles, as well as lysosomes, right? So these all together form the endomembrane system. Since they are covered with the membrane, so hence these are together interconnected. Physiologically they are interconnected, so they are interconnected among themselves. Because of this one, all these cell organelles together form what? It is forming the endomembrane system. But the question in question, GERL, G Golgi complex, endoplasmic reticulum as well as lysosomes, right? So is a part of endomembrane system which does not include the. So here it does not include what? Vacuole. So here it has left vacuole over here, right? Apparato reticulare is the original term used for. Apparato reticulare is the original word used for. See the options now. Endoplasmic reticulum, Golgi complex, mitochondria as well as chloroplast. So, apparato reticulare itself is another name for Golgi complex. It is another name for Golgi complex. So, remember this point also, right? So, and the next question is, next one is, with genus green mitochondria turn, right? See here. So whenever the mitochondria are stained over here, so with the genus green itself, that stain, that moment their color turns to what? It turns usually to greenish blue. It turns to greenish blue color, right? Next question is, the functional unit of mitochondria is, very important question it is, the functional unit of mitochondria is, means that structure must so responsible for the function of the entire mitochondria. See here, this is a double membrane bound cell organelle, that is mitochondria is there. So inside the mitochondria, the inner membrane itself makes the finger like projections over here, and on these finger like projections you will find a stalked structure there. You will find the stalked structures there, and these are known as what? Oxysomes. These are known as oxysomes or F0 F1 particles. So right, so elementary particles like this, many names are given over here for these. So stalked structures, and they are responsible for the synthesis of ATP molecule over here. Hence they are the functional units in mitochondria, right? Then the answer for this question itself is oxysome is the functional unit of what? So the mitochondria, right? Next question. Pallid granules are, already in theory I have covered this one. So pallid granules are nothing but they are simply the protein synthesis that occurs in this cell organelle, and its name itself is what? Ribosomes. So these are the protein factories. So protein factories, ribosomes are called as pallid granules. Why? Because Pallid saw them. So for the first time, and they were called as pallid granules. Okay, so myeloblast, lysosomes, spherosomes, And next one, ion essential for linking ribosomal subunits. Which ion is responsible for the unite of both the subunits over here? See here, this is the larger subunit and this is the smaller subunit. This is 30S ribosomal subunit, this is 50S ribosomal subunit. In case of 70S ribosomes, and in case of 80S ribosome, it is 40S, it is 60S. In case of 80S ribosomes. So for the union, these are basically being separate. So these unite each other in presence of one of the ion, that is nothing but magnesium ion, is too essential for their unite. Hence the answer for this question is magnesium. Right? So next question is, which is not true for the eukaryotic cell? There are some statements are given over here. Among those statements, which is not so true for the eukaryotic cell. So what is the not true statement over here? Let us see. It has ATS type of ribosomes in mitochondria. So mitochondria usually present inside the cell have their own ribosomes, of course. So they are about 70 ribosomes. See here, this is mitochondria inside which 70 ribosomes are usually present over here. So that's why the correct answer, so for this question is the first one. Why? Because so he says which is not true for the eukaryotic cell. That's why the first statement is the true for not true for eukaryotic cell. Next statements also will have to analyze at any cost. What is the next statement? See here. It has ATS type of ribosome in the cytoplasm. In cytoplasm, so this has what? ATS ribosomes. Of course, it is the correct statement. Mitochondria contains circular DNA. Of course, it contains the circular DNA over here. It is quite natural. And the next one, membrane bound cell organelles are present. Membrane bound cell organelles are present over here. So what are the membrane bound cell organelles present inside this one? So the question arises. Right? In eukaryotes it is quite natural. Membrane bound cell organelles are present over here. This is also true, this is also true, this is also true, but this is the only statement which is wrong. That's why that is the answer for this question. Next question. Plastids differ from mitochondria on the basis of one of the following features. Mark the right one. See here, so there is a difference between chloroplast as well as mitochondria. What difference that we come across? Let us analyze the answer. Presence of two layers of membrane. It is right, of course, it is true for both. The chloroplast is also double membrane bound cell organelle, and so mitochondria is also double membrane bound cell organelle. No doubt at all. It is true. Presence of ribosomes in both the cell organelles. Another cell organelle called as ribosomes are usually present. There are 70S type of ribosomes are present over here. This is also true. What about the next one? Presence of chlorophyll pigment. Chlorophyll pigment is only present in the chloroplast or the plastids, but the chlorophyll pigment is not present inside mitochondria. This is the statement that makes the difference over here. Option C is the right answer for this question. And the presence of DNA, circular DNA is present in so the mitochondria as well as chloroplast. It is quite natural. Hence these are said to be as what? Semi-autonomous cell organelles. Next question. Peroxisome is known so it takes part in. Peroxisomes are single membrane bound cell organelles which are very smaller enough and they are so spread inside the cytoplasm and they perform photorespiration. They take part in photorespiration, peroxide metabolism. So are the major functions of these peroxisomes. Let us analyze the answers over here. So it is photorespiration is the right answer for this one. Peroxide formation and breakdown is done by the same. Direct oxidation and peroxide formation is done over here. All the answers are correct, of course. So here glyoxylate cycle. It is confined to glyaxosomes, right? So hence here the question is somewhat changed. We'll have to make it. What is the? It does not take part in. If it is said so, then it is the right answer for this question. In question, little bit change we'll have to make in it. So next question I would like to go through. Total number of fibrils present in cilium and centriole are respectively. So very important question it is. We'll have to think over here. The number of the fibrils we'll have to count over here in the cilium as well as in case of so the centrioles. See here in the centrioles what happens over here is totally 9 triplets will be there on the periphery. But at the central axis, it is made up of hub. That's why total number of fibrils in centrioles is 9 into 3 is equal to 27. Right? Why? Because they are triplets. Hence, these are 27. Triplets in the sense that 3, set of 3 will be there. In case of cilium, whenever we observe, 9 are peripheral one, 9 pair, that is doublets are present on the periphery. 9 into 2 is 18. But 18 is not the final one. Central axis is made up of 2 singlets. 2 singlets in the sense that plus 2. Total number of fibrils present is 20. Then which is the correct answer? Let us see here. So it is 20 as well as 27 is the correct answer for this question. Right? So very simple one. Next one. Dynein protein with ATPase activity is present in a cilium. Of course in the cilium. So dynein protein is making the side arms over here. Right? So doublets, you know it better. So one of the doublets, so one has side arms, that is one of the outer one and another one is inner one. Outer one is hook like structure, right, that binds to the next B of the next peripheral doublet itself. So during its activity, during the movement itself. That's why answer for this question is side arms of subfibrils. There are 2 subfibrils will be there. So there one of the subfibrils has 2 arms and this is the correct answer for this question. Next one. Central fibrous lamina is present inside. Fibrous lamina in the sense that so we know nuclear lamina, which is a fibrous one, which is lying inner to the inner membrane of the nucleus. So see here, this is nuclear membrane. So inner to this nuclear membrane, there you come across a fibrous material over here, and that makes the nuclear lamina. Nuclear lamina to which? The ends of the chromatin network that attaches over here, that is binded over there. Hence it is spread across equally inside the nucleus. So that is why the correct answer for this question is fibrous lamina is present inside the nucleus. Nucleus is the correct answer for this question. Right? And so we'll go to the next question. What is the next question? Ball bearing rings are puffs occur in? Already I have told you. So these ball bearing rings or the puffs, they occur in Lamb-Rush chromosome, he says. In Lamb-Rush chromosome there are no puffs formed. Prochromosomes. Prochromosome is nothing but nucleoid, it is called as. So there also puffs are not seen. Polytene chromosomes. So these polytene chromosomes are nothing but they are simply salivary gland chromosomes. So and these chromosomes have puffs or Balbiani rings, and the name came from the scientist name, that is Balbiani itself, right? So these are some questions I have solved for you people. And next question is Lamb-Rush chromosomes are, what kind of chromosomes these are, in which stage they are seen? Already I have explained this concept during the theory. So already you know, so meiosis occurs, right? Meiosis occurs in some of the cells which are diploid in nature. This cell divides first into two cells. This is meiosis 1, it is said as, and again it divides into two cells respectively. This is called as meiosis 2, right? Meiosis 1 as well as meiosis 2, it is said as. During meiosis 1, during the formation of these haploid cells, what happens over here is, so during this one, prophase, metaphase and anaphase as well as telophase. So these are the 4 stages that you come across during the cell division, that is whether it is meiosis, whether it is mitosis, doesn't matter over here. So in meiosis 1, these 4 stages that occur, of course, during nuclear division. What happened over here? Prophase that occurred over here. But in case of meiosis 1, right, prophase has 5 stages already I have mentioned. So prophase has L, Z, P, D, D. These are the 4 stages that we come across over here. Okay, among them, so where actually this stage is seen over here. It is actually seen in diplotin. Diplotin, it is the dikinases, right? Lactotin, zygotin, pachytin, diplotin. These are nothing but diplotin chromosomes. That's why correct answer for this question is diplotin chromosomes. Lamb-Rush chromosomes are seen always in a diplotin stage. Hence they are diplotin chromosomes. They are not salivary chromosomes. Why? Because salivary chromosomes are polytene chromosomes. And prochromosomes, you know, it is another name for nucleoid or naked DNA. And L chromosomes in the sense that lactotin chromosomes. So where we come across is lactotin chromosomes. That's why L is not the answer for this question. That's why diplotin chromosomes is the correct answer for this question. Next question. Flagella of prokaryotes and eukaryotes differ in what way prokaryotes as well as eukaryotes have their respective flagellas, and there is a difference among themselves. So even though both are acting as the flagella, so there is a difference that you come across. What is the difference? So let's go through the options and search for the correct answer. See here, type of movement and placement in the cell. Placement in the cell doesn't matter, type of movement of course matters. So that's why, this option is not exactly the correct answer for this question. Next one. Location in cell, once again he says mode of functioning, right? Functioning of course differs. Microtubular organization, this is most important thing. Here microtubular organization is different and the type of movement is also different. So that's why this makes the difference between the flagella of prokaryotic cell as well as eukaryotic cell. That is microtubular organization as well as the type of movement over here. Right? That's why this is the correct answer. Microtubular organization and function, he says. Function is common for same for both the flagella. Itself is movement. So it is one of the organ for its movement of the body. Right? So next one. Foldings of inner membrane of mitochondria are called as. Many a times I have drawn the diagram in mitochondria. So there the inner membrane makes many number of foldings and those foldings are named as what? Cristae. What they are named as? Cristae. They are named as cristae. Hence here the option cristae, option C is the correct answer for this question. So it is not calcium oxalate crystals. So it is not grana. Granas are usually present in chloroplast. And next one, sacs. So sacs are not here. So that's why cristae is the correct answer for this question. Next question. Polytene chromosomes in the salivary gland of Drosophila melanogaster are formed as a result of. How the polytene chromosomes or the giant chromosomes are formed, I had explained in theory. So what happens over here, the DNA what is present inside these chromosomes, it starts to duplicate without cell division, right? So the DNA is usually present inside the nucleus, right? It is present inside the nucleus and that duplicates again and again and again and again. So ultimately it makes many replicas of the same DNA. Right? So all together form large sized chromosome there. So that large sized chromosome is formed as a result of the duplication of the DNA. So present in each chromosome there. So see here, Drosophila melanogaster has salivary glands. In those salivary glands, polytene chromosomes are seen. They are formed as a result of the replication of DNA without cell division is the correct answer for this question. What is that? Replication of the DNA without cell division. So here endoduplication, he says, duplication without separation, he says. So these are not the correct answer for this question. Next one. Which is not true about Spherosomes? Spherosomes are once again the microbodies, so present inside the cytoplasm, and these spherosomes are minute structure which are single membrane bound structures. Right? See here, the statement which is not true about the spherosomes will have to identify. See here, they arise from the endoplasmic reticulum. Many a times I have told you during the study of these spherosomes. So these are usually the pinched off vesicles of endoplasmic reticulum. Endoplasmic reticulum has cisternae, tubules as well as vesicles. Those vesicles later become what? Spherosomes. That's why option A is true. Single membrane bound cell organelles, already I have mentioned, that's why it is also true. Okay, both A and B are true here. Related to fat, right? Of course, they are fat storing cell organelles. So involved in photorespiration. So this is not a function of these spherosomes, right? So photorespiration doesn't occur over here in spherosomes. So that's why it occurs usually inside, right? Peroxisomes. So that's why, so this is not the true statement according to or confined to the Spherosomes. So that's why this is the correct answer for this question. Right? Option B is the correct answer. Next question. In which one of the followings would you expect to find glyaxosomes? Right? Glyaxosomes are usually present in those cells, especially those structures which contains especially the oil containing seeds it is present in. Example, castor as well as groundnut. So there it is present. And especially it is present inside the endosperm, right? It is present inside the endosperm. The question arises, what is endosperm? You may know the coconut itself. This is coconut you consider, and there you come across another white layer of the coconut, right? White layer you come across. This is hard and sturdy, which is made up of the dead tissue, that is sclerenchyma tissue, right? And sclerenchyma tissue it makes, and there you come across the watery substance inside it. And this watery substance is said to be as endosperm. Endosperm is the nutritive tissue for the developing embryo. In coconut, on one side, you will find this kind of bulge over here. This is nothing but embryo, right? During the growth of the embryo, what happens here? This water is completely utilized by this developing embryo, so that it can grow into new individual, right? So this is the fact what you should remember. So, and we haven't gone through the seeds this year, so that's why, so we don't know clearly about this picture, that is endosperm picture itself. So that's why, just remember, endosperm itself is the nutritive tissue for the developing embryo. So here the concept regarding this one is, so glyaxosomes. Glyaxosomes are a kind of microbodies which are present in endosperm of wheat, he says no. Endosperm of castor, he says it is oil containing seed. Where, so these glyaxosomes are usually present. Then answer is endosperm of castor. So palisade cell in a leaf, palisade parenchyma, you know it better. Root hairs. So right. So the correct answer is endosperm of castor. These are seen in. And Golgi apparatus is surrounded by. This point is very important. See here, Golgi apparatus is usually present. So it is very clear. So clearly seen over there. So why this is present? Why? Because this is present especially in one area, in a fibrous area, where it is called as zone of exclusion. What it is known as? Zone of exclusion it is called as. So it is not completely mingled with the cytoplasm. So it separates from the cytoplasm. That area it is present, hence it is said as zone of, it is surrounded by zone of exclusion. This point to be noted over here. Next question. Which of the following are responsible for red color of tomato? Already we know, so how one plastid that converts itself into another plastid. So during interconversion of these plastids I have explained there. So what is the actual pigment responsible for the red color of the tomato is, so that is phycocyanin, it is blue purple color. Pinocytosis, this is cell drinking, it is called as, it is not confined to this concept. So lycopenes, see here, lycopenes are the pigments responsible for the red coloration of what? Red coloration of the tomato. So that's why option C is the correct answer for this question. Next one. Which of the following subunits of ribosome is composed of 23S of rRNA, 5S of rRNA plus 32 proteins over here? Already I have told you and explained you people regarding what the two subunits of ribosome. See here, this is 70S ribosome whose smaller subunit is 30S, whose larger subunit is 50S. So total it is 70S. In case of 80S ribosome, what happens? It is 40. This is 60S. So here the rRNA is different over here in both the subunits. Here also rRNA is so different here. But in case of this 50S ribosomal subunit, 23S as well as 5S are the two strands of, these RNAs are usually seen over there. So that's why the correct answer for this question is 50S of 70S ribosome. So 50S of 70S ribosome we can see. That's why. So this is the correct answer for this question. In the diagram, which of the following processes are shown in an amoeba? So let us draw the diagram of the same which is given in our material. So later we'll analyze what processes are going on inside it. See the diagram now. See here, this is one amoeba where seropodial extension and this is the food. This is solid food, he says, right? And the same food that enters over here and uncovered with a vacuole, right? So that comes over here, and ultimately the same that comes over here and it mingles with another, what is that? So food vacuole, he says, food vacuole, he says. And next one is the residue of undigested food. The residue of undigested food, he says, and it is thrown out. It is thrown out of the cell. It is thrown out of the cell into the cytoplasm. This is the diagram which is given in our material, right? So see here, solid food, if it is engulfed, the engulf of the solid food and intracellular digestion itself is said as phagocytosis. It is confined to phagocytosis. If instead of solid food, if there is any watery substance or liquid, so then it is called as pinocytosis. It is called as pinocytosis. And the residual or the undigested food material that remains inside one vacuole and it is thrown out of the cell in the sense that exocytosis it is called as, right? This is endocytosis, this is exocytosis it is called as. See here, all these processes are going on over here except pinocytosis he has to mention as if here the liquid. He must have mentioned the liquid food, solid or liquid food. If he mentioned, then all the options are correct over here. See the options. In a diagram, which of the following processes are shown in amoeba? Phagocytosis, of course. So this is also shown over there. And pinocytosis as well as exocytosis. All of these are shown over here. So but pinocytosis he has to mention as liquid food. If he mentioned it, then this is also right. Then all the above itself is the correct answer for this question. Okay. Next one is very important one. Here pits are formed on the cell wall due to lack of. What is lacking over here during the pits formation? So as in the diagram you can see where the pits are formed. Let us see here. See here, this is one of the cell, hexagonal cell I am going to draw over here, and on this we will find see here, this is one plant cell what I have drawn over here. So this is another adjacent cell. What you come across here is, see here, this is called as middle lamella. This is called as middle lamella. It is a common cementing wall between. So and this one is said as primary wall. Okay. Inside the primary wall you will find the secondary walls. Secondary walls. See here, during the formation of secondary wall, you will find there is a gap, and this gap is nothing but the pit. What is this one? Pit. It is called as. Say for example, if you analyze the same thing, this is middle lamella, over it primary wall, over it primary wall, over it secondary wall that we come across over here. And here it is pit area, it is called as pit. So due to the lack of the secondary wall in certain places, so what is happening over here? So pit is formed over there, right? Hence, what is the answer for this question? Let us see. Pits are formed on the cell wall due to lack of cell plate, he says. Actually, it is not cell plate. So, primary wall material, he says. No, it is not regarding the primary wall. This is called as primary wall, right? This is called as primary wall. That's why it is not regarding the secondary wall material, of course. So, this is the correct answer for this question, right? Middle lamella, of course, it is present at the middle part of the cell wall, right? So next question is, what is the next question? See, ribosomes are attached to endoplasmic reticulum through magnesium peptidyl transferase, rRNA, ribofourin. Already we know ribofourin 1 and 2 are the specialized proteins. They help in attachment of these granular structures, pallid granules on the outer membrane of the endoplasmic reticulum. Already I have mentioned in theory. So that's why the correct answer for this question is ribofourin is the correct answer. What about rRNA? It is one of the composition of what? Ribosomes. So peptidyl transferase is one of the enzymes required for translation. As you know, transcription, in the same way, next step of the protein synthesis is translation. There are two steps that we come across. One is transcription, you know it better. biosynthesis of mRNA on one of the strands of DNA. So here second step is, so that is translation. During translation, peptidyl transferase is an enzyme required there. And the next one, magnesium. Magnesium is helpful to unite both the subunits of ribosomes. That's why the correct answer for this one is riboforine. And the last question is, the nucleoprotein structures that occur at the ends of the chromosomes are. What is the nucleoprotein? What is nucleoprotein? Nucleo and protein, right? Nucleic acids, they are associated with the proteins, hence they are called as nucleoproteins. This nucleoprotein is nothing, it is DNA as well as proteins. So combined together form nucleoproteins. So those nucleoprotein structures that occur at the ends of the chromosomes are. See here, if this is the chromosomal structure, then you will find this kind of structure there. Of course, this is one chromosome. So here, what is that? Satellite is there. And this is called as telomere. The nucleoprotein at the end of the chromosome itself is telomere, right? So that's why the correct answer for this question is telomere, right? So we solved near about 36 number of questions in this session. In the next session, I am going to solve some more questions for you people, and so after that one level 3 will solve so some bits, and after that one we will start with a new chapter. Thank you.